All right, I appreciate the opportunity to talk with you all today. So I just want to first show you that this is our institute in Florida. It's a new institute that was developed under a partnership with the state of Florida, and we are only been in our building for about a year. I am the program director of the Vaccine and Infectious Disease Division, and we are currently still trying to fill out all our recruits and going to have a nice department as soon as I get all these people hired. But what I should tell you is that the data I'm going to show you today was when I was a professor at the University of Pittsburgh. And these were done by three outstanding researchers in my laboratory. Donald Carter, Corey Krivar, and Brendan Giles are the ones who performed all this work. So one of the goals for developing an influenza vaccine is to try and overcome some of the limitations associated with cross-reactivity between different strains. As we all know, it has been a very difficult challenge to predict each year exactly what the most appropriate strain is to use for seasonal influenza, because there are so many different variants that can emerge. And so from year to year, we have to keep changing the vaccine. We wanted to address this problem, and what we first started to do, and the data I'm going to show you today, was with H5N1, and this was supported by grants from NIAID as well as Path Vaccine Solutions. But we've now moved on in partnership with Sanofi Pasteur to develop this strategy for seasonal influenza for H1, H3, and B influenza, as well as really for any subtype that is emerging, such as H7. What we are still focusing on is the hemagglutinin protein, so I'm going to show you the data associated with that. And really there's been a lot of attempts to develop a cross-reactive vaccine that will elicit a cross-protective immunity that will protect you no matter what type of strain or what type of clade will emerge. But this has been done with very limited success. So as you know, pandemic influenzas can come from anywhere, and we've seen that over the last 10 years or so with the novel H1N1, but also with H5N1. And so a variety of different companies and manufacturers are targeting different strategies to come up with this broadly reactive or universal type of vaccine by looking at conserved proteins in the virus, such as NP or M2, or looking at other molecules. We're still interested in the hemagglutinin since it's the most immunogenic molecule that elicits an immune response against the virus. And so in order to do this, we really have to come up with a way to overcome some of the genetic diversity associated with HA. Anytime there is a new pandemic strain, we do have to deal with a highly pathogenic type. It's usually novel, so how to deal with something how to make a vaccine against something that you don't yet know exists is a challenge. And of course, with looking at H5N1, the early issue is that it is yet to be able to transmit from human to human. But if it ever did, or any other subtype ever did, we would have a new emerging pandemic strain. So when we first tackled this problem in 2007, 2008, we looked at H5N1 because this was the one that seemed to be emerging and coming out of the poultry flocks. And what you can notice is there's a diversity associated with different subtypes of H5N1. So the 10 phylogenetic clades have been identified. These are geographically distinct, but of these 10, only 3 or 4 of them have been able to infect humans. Clades 0, 1, 2, and 7 have we actually isolated. But when you look at the sequence database for hemagglutinin sequences, what you'll notice is that there is definitely a bias in the number of sequences returning to a few clades, and those are mainly the ones affecting humans, clades 1 and clades 2. So but nothing says that clades 1 or 2 will eventually become the virus or the virus out of a particular clade that is able to replicate and efficiently transmit. It could come from one of these other clades for which we have very little information for. So how does one design a vaccine against a particular strain or a clade that we don't yet know exists? Well, we chose to see if our strategy would work against clade 2 isolates because these were the isolates that seemed to be transmitting and moving across the Eastern Hemisphere from China and Southeast Asia across into Russia, Middle East, Europe, and Africa. And so could we design a vaccine that would address all the different subclades and sub-subclades of clade 2 that had been infecting people and infecting wild bird populations, with the most likelihood of meaning that one of these strains would emerge and eventually become a human transmittable virus. So H5N1, as you know, has some challenges. First of all, its virulence complicates the ability to make a vaccine. Wild-type viruses do not grow well in eggs. They usually are barely pathogenic to eggs. It usually mandates a BSL-3 manufacturing, which is also a major challenge for H5N1. As has been noted here at this meeting today, H5N1 HA is poorly immunogenic. It takes large doses to elicit a modest immune response. And there's really poor cross-reactivity between different clades. And even though we heard today that there's a stockpiled vaccine that's been approved by the FDA, it really doesn't work very well against, for example, clade 2 isolates. So how does one predict what is the clade or strain that's going to emerge, and how do we increase the breadth of our immune response to H5N1 vaccines so that we can cover whatever strain actually emerges? So the real question is, how do we overcome this viral diversity? There's been a couple of ways in which broadening strategies have been used in the past for flu as well as other viruses, such as making a polyvalent mixture. And we do that currently with the flu vaccine by having an H1, an H3, and a B antigen in our vaccine each year. But another way that's been tried, it's been tried for HIV and some other viruses, is doing a centralized sequence. So can you come up with a sequence that will capture multiple different antigenic features within that sequence, which would elicit a broadly reactive immune response? It's been tried for 3 different methods. One is ancestral, one's called center of the tree, and the most common one has been consensus. And consensus is just identifying the most common amino acid at any given position and making a synthetic gene based upon what is the most predominant amino acid in the sequence. This has been limited ability to use a consensus sequence for H5. It hasn't shown to be very immunogenic. And so our goals were, how could we overcome this by making a centralized sequence that was more antigenic, more immunogenic, and the ability to elicit an immune response that would react to the diversity in clade 2. We chose clade 2 because of its diversity, because its prevalence, and because it was actually spreading, and is still spreading across the continent. We compared a centralized antigen to a polyvalent mixture, and we evaluated for protections of a mechanism of protection. So here's an example of how we generate our vaccines, our immunogens. The name that we use is computationally optimized broadly reactive antigens. This is an acronym known as COBRA. Our COBRA vaccines are actually based upon amino acid alignments against clade 2 from human isolates. This is our first attempt to generate a COBRA-based vaccine. And what we do is use a layered consensus approach in which we limit the sampling bias in the databases. And we do this by doing sequential rounds of sequence building against various clades and subclades within clade 2, as well as based upon kinetic outbreaks. So oftentimes in the database, what you'll notice is that there's a whole variety of isolates from a single outbreak or a single period of time. Well, really, we treat all of those as one single sequence instead of treating them as a bunch of individual sequences. And this is one way to limit the bias that is associated with what's in the database to generate a more accurate synthetic sequence that will elicit a broadly reactive response. And by doing multiple rounds of sequence building, we look at different subclades of clade 2, which then generate a secondary consensus sequence, which eventually gets us to our final sequence and retains the epitopes associated with the clade 2 human isolates from all of the initial strains for which we started with. If you were just to build a general consensus sequence, clade 2.1 isolates predominate in the database. So you would end up with a clade 2.1-like sequence. We eliminate that by doing this layered sequencing in which the 2.1 epitopes are retained as long as with 2.2 and 2.3 and any other ones that we put in there so that their epitopes are contained in the final sequence. What we end up with is an immunogen, an HA immunogen that doesn't fall with any of the different clades or subclades of H5. It's usually about equal distance. And what we did for our studies is we used a virus-like particle backbone. So as you've heard today, virus-like particles are very effective immunogens. They allow us to express the HA molecule in its correct three-dimensional conformation. We have to use a matrix core along with NA to get release of our virus-like particles. These can be made from mammalian tissue culture, from baculovirus systems, from plants. And what we're going to show you today is all of ours were made from a mammalian cell culture system. So the very first study we ever undertook using our COBRA-based antigen was to express it on the surface of a virus-like particle. Compared that to another HA that was derived from a wild-type strain, a clade 2.2 isolate that was isolated in 2005. Those two vaccines were compared head-to-head in a mouse system in which the mice were vaccinated at week 0 and again boosted at week 3. And then these were challenged with highly pathogenic H5N1 representing the exact virus that was matched to the clade 2.2 isolate. What I'd first like to do is direct your attention to panel A. This is looking at an endpoint dilution titer in ELISA. The black bars represent the COBRA-based vaccine, the white bars represent the clade 2.2, and the mock vaccinated are shown in gray. And what we're looking at is the amount of antibodies that were elicited to the HA molecule, to a clade 1, a clade 2.1, a 2.2, or a 2.3 isolate. And what you'll see is regardless if the animals were vaccinated with a COBRA-based vaccine or the wild-type 2.2 isolate, they elicit a high titer antibody that recognizes all different versions of HA. However, if you look on the right-hand side, when we're looking at HI titers, which are receptor blocking antibodies, you'll notice that the COBRA-based vaccine recognizes all of the clade 2 isolates, and they recognize the clade 1 isolate we used. But the wild-type clade 2.2 does a poor job, but does recognize its own clade 2.2 isolate, but doesn't recognize clade 1 or the other clade 2 isolates. So we're actually making an HI, and we've also shown this by microneutralization, neutralizing antibody titers that are broad across a variety of different isolates representing different clades and subclades of H5, even though they all have very high titers. Of course, when you challenge these animals, the animals are all protected. The COBRA-based vaccine protects just as well as the wild-type based vaccine against its own clade virus that's used for challenge. We then look to see whether or not the COBRA-based vaccine was able to do as well as a polyvalent mixture. So we actually made a clade 2.1 isolate, a 2.2 isolate, and 2.3 isolate, all wild-type HAs, and mixed those together in a polyvalent mixture, and then vaccinated the mice and to see whether or not they actually made a broader response than the COBRA-based vaccine. And what I direct your attention to here on the right-hand side is, again, HI titers, showing that the COBRA-based vaccine was able to recognize all the different subclades of clade 2 as well as clade 1, but the polyvalent mixture was able to recognize each of the individual clade 2s since they were included in the vaccine mixture, but not at the same height or titer, and wasn't able to recognize any clade 1 isolates. We challenged these animals with highly pathogenic H5N1, and what I'd like to direct your attention to is the fact that the COBRA-based vaccine, shown here in black, is able to eliminate the virus from the lungs of these animals very quickly, compared to the wild type, which took an extra couple of days to eliminate the virus from its system. We see the same response whether or not you're looking at individual or polyvalent mixtures. The COBRA-based vaccine is able to eliminate the virus within about 1 to 2 days from the lungs of these animals, whereas the other strains actually take 3 to 5 days to eliminate the virus from the lungs. We see the same phenomena when you challenge with the clade 1 isolate. This is the Vietnam 2004 strain. Again, the COBRA-based vaccine is able to knock down the virus in the lungs of these animals very quickly, whereas the polyvalent mixture in this case, or individual viruses representing different clade 2, are unable to do it in less than 5 days. I want to say that we recapitulated all of these results in ferrets, looking at nasal washes and looking at the lungs of these animals, and just due to lack of time, I'm not going to show you. They're very repetitive, but it's exactly the same results. What we did do is undertake a monkey study in which we took Somalis macaques and vaccinated them with our VLPs expressing the COBRA-based vaccine or this wild-type clade 2.2 HA. The monkeys were vaccinated twice, at week 0 and week 3, and then at week 9 were challenged with the highly pathogenic clade 2.2 isolate. And what I'd like to direct your attention to is first look at the middle. We're looking at HI titers against a whole panel of H5N1 isolates. And so if you look right here in the center, viruses representing clade 2.2, which match the wild-type strain, show that the COBRA-based vaccine recognizes all of these isolates very well. The 2.2 isolate recognizes most of them, but not all of them. And as you move outside of clade 2.2, you'll notice the COBRA-based vaccine recognizes many more isolates outside of across clade 1 and even up to clade 7, whereas the wild-type strain is only able to recognize clade 2, a few of the clade 2 isolates outside of 2.2, and really doesn't recognize anything outside of clade 2. So we get a broader breadth of our antibody response, recognizing a whole panel of H5N1s, and they represent not just from the same era that they were isolated, 2005, for which we made our use the sequences to make our vaccine, but we recognize 2007 isolates and 2008 isolates. And so some data I don't have on the slide is now we have viruses from 2013, and the antibodies are still recognizing 2013 isolates. So we have a long period of time for which we're recognizing these strains. Several of these monkeys were sacrificed at day 3 post-infection, and we ran H&E stains on their lungs as well as antibodies to detect virus in their lung tissue. On the very right-hand side, the non-infected animal shows a really healthy lung with lots of airway visible. On the very left-hand side, animals that were mock vaccinated and then challenged with H5N1 show a large infiltration of cells and granulocytes into the lungs of these animals. There's a lot of condensation, and you'll notice that there's a lot of virus that is detected here in these brown spots. When you compare the two vaccinated animals, whether they got the wild-type strain or they got the Cobra-based vaccine, the animals that were vaccinated with the Cobra VLP actually have lungs that look very similar to non-challenged or non-infected animals. There's lots of airway space, and really we were it was very difficult for us to find any virally infected cells at day 3. This is about the only one we were able to find. The wild-type strain, while it doesn't look as bad as the mock-vaccinated animals, definitely have a lot of cells in their lung. They have high viral titer that's able to be detected here in their tissue as well as in their washes and bronchial lavages that we performed. And so these animals were definitely ill and sick, and they succumbed to the disease. So what I've been able to show you today is that these Cobra sequences are functional proteins. They bind to the receptor, they bind to HA or red blood cells. The Cobra vaccine is immunogenic. It has the ability to protect against viral isolates from a whole variety of H5N1 clades and subclades. All the vaccinated animals that we vaccinated were completely protected from morbidity and mortality with Cobra-based vaccines. And these animals had decreased viral replication in their lungs compared to animals vaccinated with wild type. So we've expanded these results to a second generation of H5N1 in which we've included avian isolates. We've developed these vaccines for seasonal influenza in partnership with Sanofi, and we now have data coming off in this quarter for the H1N1 showing that we have a wide breadth of H1N1 reactivity, which we should be able to show at some of our future conferences. And we've been able to capture multiple epitopes in a single immunogen to elicit this broadly reactive polyclonal antibody response to the HA head. We don't have a ruling out that we're not also eliciting responses to the HA stem protein, but we do know that we're reacting to the HA head. So we're hoping that this work can be translated to seasonal influenza, not just H5N1, and we should know that soon. So acknowledgments for people from my laboratory at the University of Pittsburgh was Corey Crevard, Donald Carter, and Brendan Giles. Really is the ones who showed you the data today, as long as the rest of my laboratory over time that has worked on influenza projects. We work closely with Clayton Wiley in the Department of Neuropathology, and he ran all of the H&E and all of the stains that you saw today. And we do work with a whole variety of different groups around the world, and this research was funded by NIAID, by Path Vaccine Solutions, and by Sanofi. I'll take any questions you have. Thank you.